Chapter 3, Topic 3.3, the cell envelope, the wall, and membranes. In this topic, we will be discussing the cell envelope of bacteria. Specifically, we're going to talk about what the cell envelope is, differences in cell envelopes among different prokaryotes, the cell wall, the gram-negative outer membrane, and cytoplasmic membrane structure. At the end of this recorded topic lecture, you should be able to meet the following objectives. Differentiate between the two main types of bacterial envelope structure. Discuss why gram-positive cell walls are stronger than gram-negative cell walls. Know the steps of the gram stain and know why gram-positive cells stain purple and gram-negative cells stain red. Name a substance in the envelope structure of some bacteria that can cause severe symptoms in humans. Now let's talk about the cell envelope. The cell envelope is the complex external covering of the bacterial cell. It is composed of the cell membrane, the cell wall, and in some bacteria, the outer membrane, specifically in gram-negative cells. We're going to be talking more about each of these as we go through this topic lecture. Back in the 1800s, a Danish physician named Hans Christian Gram discovered that different groups of bacteria stained in different ways when he applied a special stain, which was eventually named after him. This was because of differences in the cell envelope. Think I can work difference into this again? Of course, we didn't know then the different reactions to the gram stain were due to differences in the cell envelope. Yep, I did. Now let's go over the steps of the gram stain and talk about why we get a different reaction in gram-positive bacteria versus gram-negative. The first step in the gram stain is to apply crystal violet, which is, as the name implies, a violet or purple color. This stains the cell walls of both types of bacteria. Next we add Gram's iodine, which is a mordant or a chemical that forms a complex with the dye. So you can see over here in our magnification of the cell wall that we've got bigger pieces of stain, so to speak. Now it's more firmly trapped in the thick peptidoglycan cell wall of gram-positive bacteria. Over here it's not as effectively trapped because gram-negative bacteria have this outer membrane. Then we apply alcohol, and alcohol doesn't have any effect on the crystals in the gram-positive cell wall, but this outer membrane is kind of broken up by the application of alcohol. Alcohols break up oils, so the stain leaves the cell wall of gram-negative bacteria and they become clear again. They lose the purple stain. Now we could stop there. We could just have purple gram-positive bacteria and we could have clear gram-negative bacteria, but clear cells are hard to see under bright field microscopes. So we add our secondary stain, safranin, which is a red stain, to stain the clear bacteria. Now the red dye is masked by the violet. Here's another way to think of it. This peptidoglycan in gram-positive cells are already stained, no room for a second stain. But the red stain does stain the peptidoglycan cell wall of gram-negative bacteria, turning them red. So if we do a gram stain properly, and if there's a mix of gram-positive and gram-negative cells on the slide, we will see purple and red cells on the slide. Now make sure that you know the gram stain procedure and what gram-positive and gram-negative cells look at each stage and why they look like that at each stage. It's important to understand the structure of cell envelopes and why the gram stain can tell the difference or differentiate between the two types. All but a few bacteria have a cell wall, so we'll start there. The cell wall of all bacteria is composed of a combination of carbohydrates and amino acids called peptidoglycan. Peptidoglycan is only found in bacteria. Now in the textbook it has a diagram showing a better representation than what I'm going to show you here, but this gives you the basics of the composition of peptidoglycan. So glycans, which are sugars, actually they're altered glucoses, are hooked together to form glycan chains. Now the glycan chains are hooked together by short chains of amino acids. Some of them have 4 amino acids, some of them have 7. And this holds everything together to form peptidoglycan. Now, don't worry about the exact amino acids and glycan units, but you will need to know the basic structure. Here is a diagram of a gram-positive cell envelope. Notice the thick peptidoglycan cell wall. Also notice the lipoteichoric acid, which is unique to gram-positive bacteria. Acid-fast bacteria have a thick cell wall and will stain weakly gram-positive. They are genetically related to the gram-positive group, but their cell wall is also composed of mycolic acid, a wax. This makes it so that it's hard to stain them using the gram stain. Think of trying to stain an Easter egg that you've written on with crayon. The stain doesn't attach to the crayon. This mycolic acid also contributes to their disease-causing abilities in humans. Because of this waxy surface, they require a special stain, the acid-fast stain, to identify them. Here is a diagram of a gram-negative cell envelope. Notice the thin peptidoglycan cell wall sandwiched between the outer membrane and the cell membrane. Also notice the lipoproteins. They're known as lipopolysaccharides, or LPS, which is unique to gram-negative bacteria. Now on to the gram-negative outer membrane. Now you'll notice that the gram-positive cell envelope ends with the thick peptidoglycan cell wall. Over here on the gram-negative side, you'll see that we have the thin peptidoglycan cell wall, then we have this outer membrane. And it's similar in structure to the cell membrane, which we will be covering later. Now, as we mentioned in the last slide, the outer membrane contains LPS, or lipopolysaccharides, which are referred to an endotoxin because it is inside the cell, it's not released by the cell, and it stimulates fever and shock in some gram-negative infections. The outer membrane also allows only very small molecules through. This makes gram-negative bacteria naturally resistant to some antibiotics. Onto the cytoplasmic membrane structure. The cytoplasmic membrane, or cell membrane, is a phospholipid bilayer. Remember that phospholipids have a water-loving or hydrophilic head and water-fearing or hydrophobic tails. The heads naturally face toward the water-based solutions outside the bilayer, and the tails face toward each other. The membrane also contains proteins. Okay, see, there are the proteins in both the gram-positive and the gram-negative. These proteins help molecules move in and out of the cell, sense the environment, and so on. In most bacteria, the cell membrane molds to the cytoplasm inside of the cell. In some bacteria, though, the cell membrane forms folds on the insides of the cell. This provides greater surface area, or room, for enzymatic functions such as photosynthesis and aerobic respiration. This leads to the function of the cell membrane. It provides a place for some processes, like photosynthesis, to take place. It also regulates what comes in and comes out of the cell. Well, that's it for this topic. You should now be able to differentiate between the two main types of bacterial envelope structure, discuss why gram-positive cell walls are stronger than gram-negative cell walls, know the steps of the gram stain and why gram-positive cells stain purple and gram-negative cells stain red, and name a substance in the envelope structure of some bacteria that can cause severe symptoms in humans.